Good morning, students. I, Subita Pal, and my friend Dr. Bhavini Shah, both of us are from RG Shah Science College, Vasna, Ahmedabad. Now we have come here to discuss the topic biosensors. We will try to explain you what biosensors are, what is the principle involved, what are the applications, etc. Now to start with, it is very important to understand what biosensors are, right? So as the word implies, biosensor means bio plus sensor, right? So that means this is the system or this is the device in which biological substance is used for sensing the presence of any other substrate, right? For example, you want to analyze glucose in the blood sample, right? So glucose will be considered as the analyte. So what is analyte? That is a substance which we want to analyze, right? And biological material will be any substance which is of biological origin. It can be enzyme, it can be antigens, it can be antibodies, it can be microbial cell itself, right? So what is done that in biosensor, the biological material is immobilized, right? And the analyte, now you know what is analyte, the substance which we want to analyze, right? So the analyte will react with the biological substance, and as a result a sort of signal will be produced. This signal can be anything, it can be mechanical, electrical, any type of signal. So this signal will be produced which can be measured, and this will give us the idea regarding the concentration of the analyte, right? So I hope you understand what are biosensors. So I mean, just to explain it scientifically, so what are biosensors? They are actually the biological material. It is a biosensor is an analytical device containing an immobilized biological material which can interact with an analyte and produce physical, chemical or electrical signal that can be measured, right? Now these biosensors are becoming very popular, but it is very important that we should know that how this concept of biosensing came. So the concept of biosensing basically came by the hard work of Professor Leonard Clark, and he devised the electrode for sensing the concentration of dissolved oxygen in the blood of patients who were undergoing surgery. He tried to make a biosensor which could analyze the concentration of glucose in the blood, but he could not succeed. But on his concept only, the blood glucose biosensor was later on launched in the year 1987, and after that lot of work was done in this field. Now another point is that what is so important about biosensors, why these biosensors are becoming so popular. So in that case, the first and the most important thing is that they are very small in size. Biosensors are small in size, and because of that you can easily handle them. I mean, they are very, you can take very small volume of the analyte for analysis. Another thing is because they are small, so they can be used inside the catheters or inside the needles, and they are so compact that you can use them for in the fields also. Second point is that very fast measurement is possible in this case, because you just imagine if you have to analyze your blood glucose level, you have to go all the way to the laboratory to get the test done. So it is quite time consuming, whereas in biosensors it is very fast and waiting time is very less. Within 30 seconds you can get the results of glucose by using glucometer. Then another thing is that in this case you can measure the amount of analyte in a complex mixture. Complex mixture, suppose you have blood. In the blood you want to analyze some antibody contents, you want to analyze some sort of enzyme, so you don't have to separate that enzyme, right? So in this case you can use the same complex mixture, and out of that complex mixture your desired product can be analyzed. So that is a very important positive point of biosensors. And another thing is that there is no preliminary preparation required. You can use the sample as it is for analysis. Then these biosensors, they have targeted specificity. That means that in this case the analyte which you are trying to analyze will be specifically analyzed. For example, you want to know that how many what is the titer of the antibodies in the serum. So what will you do? You will select a biosensor in which the specific antigen, corresponding antigen is there. So since the biosensor has got antigen, so naturally it is going to react with the specific antibody. So you can analyze the targeted substrate by using these biosensors. Then it is an electronic processing device, so data can be processed and stored. And another important aspect is that in this case continuous monitoring is possible, continuous measurement of the analyte is possible. So it can be utilized in the medical field as well as in the industrial processes which are running continuously. So continuous monitoring is required, which is possible with the biosensors. So now we understand what are the advantages of biosensors, right? So any biosensor which we want to select, then it should have certain important characters. It should have certain important characters, right? So the important characters we have listed, like it should be sensitive, it should have low detection limits, cost should be low, simplicity, reliability. So these are some of the required characters as you can see in the slide. So any biosensor which we want to make, it should have these characters, but it is not possible to get all the characters in a single biosensor. So we try to accommodate maximum possible characters. Now the point is that when we talk about biosensor, just now I told you that these biosensors will be able to estimate the concentration of the analyte, and I said that a sort of signal is produced. So how that signal is produced and how it is processed, that is very important to understand. And for that matter, we have to understand the principle of biosensors. But before going into those details, I'll just give you a simple example. We are actually example of a biosensor. Now, suppose a cup of tea or a cup of coffee comes in front of your eyes and your eyes are closed. So you cannot say it is tea or coffee. But still you can find it out by the aroma of the tea or coffee. So how do you know that? In that case, what happens, as you can see in the slide, suppose a cup of coffee or tea has come in front of you, you inhale the aroma, and when you inhale it, so what happens, the olfactory membrane of your nose will be activated, right? And this olfactory membrane of the nose will send the signal to the olfactory nerve cells, right? That these are the fumes of coffee or these are the fumes of the tea, right? And these olfactory nerve cells will then send the message to your brain, and your brain will tell you that, okay, in this cup tea is present or coffee is present, right? So this is the basic thing, how you come to know that in the cup contains tea or coffee, right? So in this case, what has happened, the signals have been transmitted from the coffee to your brain. Similarly, in this case, you can see that sample, you can compare it with the coffee, cup of coffee, right? So sample or analyte will react with the biorecognition element, that is biological substance, and then it will send the signal to the transducer and ultimately to the signal processing device. Now with this diagram it will be more clear. You can see that the substrate, that is the analyte, it reacts with the biological membrane, right? It reacts with the biological membrane, and as a result, any biological substance will be there. It can be, as I have already said, it can be anything, antigen, antibody, enzyme, anything, right? So it will react with it, and as a result of this reaction, what will happen, that some sort of signal will be produced. This signal can be heat, if it is some enzyme which is involved in exothermic reactions. It can be electrons, if it is some sort of oxidoreductase enzyme is involved. Some light output will be there, gases will be there, ions. It can be any type of signal, right? So what has happened, the substrate has reacted with the biological material which is immobilized, and as a result of that, a sort of signal is produced. Now this signal will be captured by the transducer, right? So transducer is a physical device which will convert this signal into the electrical signal, right? So with the help of transducer, the signal which was actually formed by the reaction of biological material with the analyte, right? So that signal will be then converted to electric signal by transducer, right? And from this transducer, this signal will be then by the help of amplifier, it will be amplified and then it will be sent to the data processing unit where it will be recorded in a user friendly manner. So that is how biosensors work. I hope it is clear. So just to make it more clear, so what is the thing? That our substrate has reacted with the biological material. As a result, some sort of signal is created. This signal will be recognized by the transducer. It will be converted into electrical signal. It will be amplified and then it will be recorded by the data processing unit, right? So now we know this is the principle. So what components should be there in the biosignal, right? Before that, just to make you more clear, biological material reacts with analyte. Bound analyte is there. It will send the biological, it will induce biological response followed by electronic response, which can be measured, right? Now we should understand what should be the components of biosensors. So first thing is we need immobilized biological element, right? So this biological element will recognize and interact with the analyte to produce a signal. Second is physical component, which is transducer or amplifier, and third one is electronic or signal processor unit, right? Now these are just figure of components of biosensors. Now depending upon the signal which we are getting, right, accordingly we have to devise the biosensors, right? So basically there are different types of biosensors. They can be electrochemical biosensors, optical biosensors, acoustic biosensors. Acoustic biosensors are also known as piezoelectric biosensors, calorimetric, nucleic acid, microbial sensors and immunosensors. Now coming to the electrochemical biosensors, they are basically sending electrochemical signals. So they are of three types. They can be amperometric, potentiometric or conductimetric. Now amperometric biosignals are generally they involve oxidation reduction reactions, because of which substrate or the product will be responsible for transfer of electrons, right? And these electrons, they will be, when these electrons flow, they will be responsible for the flow of the current, and the current which is flowing, that will be directly proportional to the substrate concentration. So just by measuring the current, you can easily find out the concentration of the substrate. This is the figure of photograph of glucometer, which is a type of amperometric biosensor. Then second one is potentiometric biosensor. Now in this case, changes in ionic concentration are determined. So potential difference between the potentiometric electrode and reference electrode can be measured, which is proportional to the substrate concentration. Examples are pH electrode, carbon dioxide electrode or ammonia sensitive electrode. This is a very interesting example of potentiometric electrode, ISFET, that is ion selective field effect transistors, which are generally used for monitoring intramyocardial pH during open heart surgeries. Then we have conductometric biosensors. Now in this case, the due to the reaction, whenever the ionic species change, so naturally it will result in the change in the electrical conductivity, and this electrical conductivity can be measured, right? So in this case, the best example is urea biosensor, which is used to detect the onset of kidney failure or malfunctioning of the livers. In this case, you can see that urea by the action of enzymes. So what will be the biological material which is immobilized? In this case, it will be urease, right? So this urease will react with urea, and as a result of that, ammonia and carbon dioxide is produced. It can be measured by either using ammonia sensors or carbon dioxide biosensors. Then we have optical biosensors. The next category is optical biosensors. Now they determine the changes in light absorption between the reactant and the product of a reaction, or we can measure the light output by a luminescent process. Now in this case, various types of biosensors can be used. The best example is LUCIFERASE enzyme based optical biosensors. Now you all know that LUCIFERASE enzyme is present in firefly, and this LUCIFERASE enzyme is responsible for emitting the light, and that is why these fireflies, they glow, right? And for this reaction, ATP is required, right? So what is done in this case? Suppose you want to find out the number of microorganisms in the any food sample, right? So artificially the bacteria are lysed. When bacteria are lysed, ATP will be released, right? Now for the reaction, which reaction? For the reaction of luciferase, ATP is required, right? If more organisms are present, more ATP will be there. If more ATP is there, more enzymatic reaction will occur, right? And because of that, more light will be there, right? So these optical biosensors are based on this principle that light, if it is emitted, it can be recorded. If less light, less organism. If more light, more organisms, right? So this is called luciferase enzyme based optical biosensors. Then we have the second type of biosensors, which are again optic biosensors. These biosensors are generally used to measure the amount of lactate in a sample, right? Now in this case, it is basically in this case what we are using, we are trying to use the principle of quenching effect of oxygen on the fluorescence of the fluorescent dye, right? The reaction, as you can see in the slide, reaction is that lactate in presence of oxygen is converted to acetate, carbon dioxide and water, right? That means if more lactate is present, then what will happen? More oxygen will be utilized, right? So if more oxygen is utilized, then available oxygen will be less. So if available oxygen is less, then what will happen? The fluorescence will decrease, right? So because we know that fluorescence will increase as the oxygen concentration increases. That is the basic thing, right? Now if lactate is more, then what will happen? More oxygen is required for the reaction, and if more oxygen is required, then available oxygen is less, and if available oxygen is less, then what will happen? The fluorescence of the fluorescent dye will decrease, and this is proportional to the concentration of lactate. So that is how fiber optic lactate biosensors are used. Then the next is the next type of biosensors, which are known as piezoelectric biosensors, right? Now piezoelectric biosensors, we also call them as acoustic biosensors. Now these biosensors are actually, what happens that there are special type of crystals which we call them as piezoelectric crystals, right? And these piezoelectric crystals, they have positive and negative charge, and they have a natural frequency. They will show the natural frequency, some definite frequency, right? Now what happens that if on the surface of these piezoelectric biosensor some small molecules are adsorbed, then what will happen? The frequency of vibration of these crystals will change, right? If the concentration of adsorbed material is more, change in frequency will be more, and this change in frequency can be recorded. So this is the principle of using piezoelectric biosensors, which are also known as acoustic biosensors. So this is the photograph of piezoelectric biosensor, where you can see that on the top of it there is piezoelectric crystal. Over the surface of this crystal, our analyte will be adsorbed, which will result in change in the oscillations, which can be recorded. And these biosensors are very commonly used to detect the presence of cocaine by using the cocaine antibodies. So in forensic science, these biosensors are being used. Then the next type of biosensors are calorimetric biosensors. Remember, calorimetric, not colorimetric. So it is these biosensors are based on the exothermic reactions. You know that there are many enzymes which will be responsible for catalyzing the reaction in which energy is, or the heat is generated, right? So if you see the difference between the temperature of the substrate and the temperature of the product which is formed, if you see the temperature variation, then it can be correlated with the concentration of the substrate, right? So these calorimetric biosensors are based on the exothermic reaction. For example, you can see here that there are certain reactions in which heat is generated. The best example is cholesterol oxidase. When it reacts with cholesterol, you can see that 53 kilojoules of energy is released. So if this energy is released, then naturally you can use this reaction for estimation of cholesterol by using this calorimetric biosensor, where you can find out how much energy is released by conversion of how much amount of cholesterol. So there is a direct correlation between increase in temperature or amount of energy released and the concentration of cholesterol present. This is just a photograph of calorimetric biosensor. Then comes very important biosensors, which are known as nucleic acid biosensors. Now you know that this is the era of genetic engineering and biotechnology. So in this case, a lot of work is being done on nucleic acids. So in this case, suppose you want to synthesize some oligonucleotide sequence, right? So this oligonucleotide sequence which you want to synthesize artificially, probably you want to use it as a primer in PCR reaction or in any other reaction. So you want to synthesize an oligonucleotide sequence. Now how will you identify that yes, the desired oligonucleotide sequence has been synthesized? So for that, what will you do? You will select a biosensor which has got complementary base sequence on the biosensor itself, right? So complementary base sequence has been immobilized on the biosensor, right? You have a fragment, let us say 20 nucleotides fragment or 40 nucleotides, whatever it is, right? So that fragment is immobilized on the biosensor, right? Now you have synthesized the oligonucleotide sequence, so you can use this biosensor. Now if this biosensor comes in contact with the complementary strand of your synthesized oligonucleotide, then what will happen? Hybridization will occur, right? The two molecules, there will be hydrogen bonding, and so hybridization will occur. So two strands, they will join together. Now how will you find out that yes, the two strands have joined? Right? So that is, you can use some fluorescent labeling technique, you can use some fluorescent dye with the synthesized oligonucleotide strand, right? So when this synthesized oligonucleotide forms a double helical structure with the nucleic acid of the biosensor, then you can say that yes, this oligonucleotide is perfect, it is the desired type of oligonucleotide. So this is the basic of biosensors, right? Nucleic acid biosensors. You can use DNA, you can use RNA, whatever you want to use. Basic principle is that it is based on the formation of hydrogen bonding between the complementary strands. So applications are that they are used for the detection of selected oligonucleotide sequences, detection or presence of mutant genes associated with inherited human diseases. They can be employed to obtain early and precise diagnosis of infectious agents, about which Madam will be talking. Now the next is whole cell microbial biosensors, that there are certain chemicals or pollutants which you want to find out. For example, if there is environmental pollution, you are dealing with that. So some chemicals are there, pollutants are there, you want to find out the concentration. You can analyze it by chemical method, but in that case you will not know the toxic effect of that pollutant. You will not understand the toxic effect of pollutant. You will not understand whether it can be degraded by microorganisms or not, so whether it is biodegradable or not, and if it is biodegradable, then whether it can be used for the organism can be used for the bioremediation or not. So for understanding all these things, it is very important to use a microbial system. Chemical analysis will not give you this idea regarding all these points, right? And that is why these biosensors are whole cell microbial biosensors are used, because they will give you idea regarding the bioavailability and whether they are biodegradable or not. And these whole cell biosensors, they are very important, lots of advantages. They are simple and inexpensive. They provide information on toxicology, mutagenic effect of the chemical, and it also helps in studying the gene expression and physiology of bacteria. And whole cell transcriptional biosensors, which are very popular, in which the pollutant, generally pollutant based promoter is there, and regulatory gene will be responsible for production of regulatory protein, and this regulatory protein will send some signal with the help of which it can be identified. It will be more clear with the next diagram. Suppose you have ordinary E. coli, right? This E. coli, it is very difficult to identify the E. coli in a natural environment. So suppose in this E. coli you are introducing the luciferase gene, So what will happen? It will acquire the ability to transmit light, fluorescence, right? And so if the culture, you have, what have you done? You have introduced the desired gene. The luciferase gene is actually the, you can say it is a reporter gene. And because of this reporter gene, it will produce luciferase enzyme, which will be responsible for luminescence. And so this E. coli can be very easily, you can very easily identify this E. coli, whereas this E. coli in which luciferase gene is not present, that cannot be identified by visual observation. So by using such reporter genes, we can use bacteria as biosensors. This is a very good example of this one, where the Photobacterium phosphorium is a type of bacteria which will emit light if it respires properly. If respiration is not proper due to presence of toxic substances, the fluorescence will decrease. And here you can see, here it is, fluorescence is there, and here the fluorescence has decreased due to the presence of toxic substances. So that is regarding the these microbial cell biosensors. Now next is immunobiosensors, about which my friend Dr. Bhavini Shah will be discussing.
Thank you, Subita. Actually, Professor Subita Pal has very well explained to you how the whole system works, how biosensors work. She has explained to you the principle, the use. Now just getting the basics of it. If you see in your day-to-day life, let's say you have visited a mall, okay? So when you go to a mall, instead of steps, you see in all high-tech malls today, you have escalators. So when you stand by the escalators, they are actually not working, but when you go close by, they start working. Or if you go to bigger hospitals, what do you see? When you go near the hospital, the glass doors are closed. But as soon as you reach the door opens. So how does this open? There is something that senses it. This is what Professor Pal had explained to you about the cup of tea and coffee. So the simple thing is that there is something which senses an event, whether it is climbing up on an escalator or whether it is the door. That sensor is somewhere where it can locate you. So when you come right at that position, it will locate you and it will open and you walk inside. Now these were all mechanical sensors. So when you talk about sensors in biology, sensor is a sensor, it senses, and we have prefixed it with bio, that is biosensors. Sensors which we use in biology are termed as biological sensors. So you have seen so many different applications that Professor Pal had explained to you very nicely. We will talk about what are these biological sensors when they are used related to immunology or related to different tests which are used for medical applications. So these are termed as immunosensors, as you have seen, where you have a flat wall. We will see in the next slides to come. We very well know that immunology deals with antigen and antibody. You have two things, antigen and antibody. So specific antigen and antibody will bind with each other. We have been learning this in TYBSc, that antigen and antibody will not bind if they are not specific. But we cannot see them. We cannot see the binding of the antigen and antibody. And therefore, you have a sensor in there. It could be an enzyme, it could be phosphatase. There are n number of sensors. We are touching the basics and the principle for you to understand. So think about this, an antigen, an antibody, a sensor which senses the antigen and antibody and gives you a color. Very, very simple. If we go to the next slide, which is a diagram, I think it will be easier for you to understand that. You see the bottom, which you see, the green, dark green, that could be a solid phase. Here it is termed as the recording device or the transducer. On that you can see the antibody receptors. So specific antigen can come in and bind to that. Let us take an example. Let us take the example of even viruses. Viruses are very, very difficult to grow on culture. Therefore, you will see that biosensors are mainly used for those fastidious bacteria or viruses which are difficult to grow in vitro on culture medium. We can grow a E. coli, we can grow a salmonella, we can grow a staphylococcus, but it is very, very difficult to grow a legionella. So in that case, when you use biosensors, it becomes very sensible, very easy and important for the diagnosis of the patient. So you see a flat solid phase on which you have coated antibodies, as you see here. These could be antigens also. When specific, if you use serum, you will have antibody inside. So if antigen is coated and antibody from the serum combines with it, we cannot see it with naked eyes. So you have a substrate and a conjugate which is added, and therefore the name comes enzyme linked immunosorbent assay, that is ELISA, whereby they combine together, and this biosensor which is present in that substrate will change the color if the antigen and antibodies have linked to each other. So if there is linkage, the conjugate in the presence of the substrate, which is the biosensor, it is sensing. Remember this, it is like the door opening in the mall. It is sensing that the antigen and antibody have combined. So if they have combined, it will change the color. Darker the color, as madam had explained to you, darker the color, more is the combining or the attachment of the antigen and antibody. It is as simple as that. That the sense or the visual look that you see is in the form of the sensor that is present in the substrate. Now this basic fundas have been very well used in the field of diagnostics. It is used in both ways. It is so interesting. It is like magic. We saw Ritik in Gujarat doing magic, but we are doing that every day. See how beautifully they have used this technology to give a very early diagnosis and which can save n number of lives in the medical fraternity. Now if you see, can I have the next slide? If you see this, these are just plain examples for you. In pregnancy test, the card test that you do at home, it has become so simple, you don't have to walk. As madam told you about glucose, you have these card pregnancy tests which give you, which has a sensor which shows you present of the beta HCG, and glucose monitoring device, as you have already seen. These are different applications. Now these are specific. If you see non-specific, see the diagram again. You have the substrate, you have the antibody, you have the purple colored squares. You see that? They are all the antigens, and the red ones you see on top are non-specific antigens. So they are not going to bind. The ones that have bind are only specific antigens. And this color will be shown with the help of the conjugate and the substrate. So the important aspect is that these are all in vitro tests. All these applications are used in two ways. They are used in vitro also and in vivo also. As you see the in vitro test, see very carefully the slide in front of you, that when we talk about diagnostics, the best advantage that the diagnostic industry has had with biosensors is that they have removed water. If you relate to any biochemical testing, what do you remember? Your chemistry labs, for example, that is what you are exposed to. If you see your chemistry lab, it is all reagents. Can you imagine working in a chemistry lab without any reagent? It sounds funny, doesn't it? But this is what biosensors have given us. Dry cassettes are available today along with the biosensors inside. And how does that help you? The main reason for a problem in reporting of diagnosis is water. In all in vitro diagnostics test, water is the culprit, because there could be a change in the pH of the water. There could be some contamination in the water, and that eventually affects the result of the test. And therefore, when you have a dry reagent, then all the problems that the water causes in the test are removed, and the results are very, very sensitive and specific. So this has been a big help to the diagnostic industries as far as dry reagents are concerned. And for consumer, of course, we see our grandparents, our parents at home who are diabetic and who had to go all the time, class 1, class 2 diabetes. For those children who are juvenile diabetic, it was so difficult for them to go to the labs and give their blood again and again. And now it's so simple. You have a glucometer at home. So all you do is, whenever you think you are feeling hypoglycemic or you feel you are feeling hyperglycemic, all you do is take the instrument and in 30 seconds you know what is the approximate level of glucose in your body. So that has been a very, very good aspect for the consumers. That is, people who are not medical people, who do not understand the medical aspect. And third, the future, of course, fantastic future it has to help mankind and a lot more. So when you see that in vitro and in vivo, all these biosensors have two gross aspects. In vitro we have discussed. When we say in vitro and in vivo, let me once again just brush up and polish up your theoretical aspect. In vitro, when we say in vitro, it is outside of the body. And in vivo, that is inside of the body. We can do anything outside of the body because it is not going to harm our body. If we have a wrong test done, we can repeat the test. But when we talk about in vivo diagnostics, it is inside of our body. Now remember this, when anything is going to happen inside our body, like ma'am showed you a nice biosensor which is used during cardiac surgeries. So these things are going to remain inside our body. Think about this, you are walking around doing your day-to-day work and those sensors are inside of your body and they are doing a job, which is a job you cannot imagine. There are n number of applications. Let us just discuss this one, which is something like an artificial pancreas. All of you know what is diabetes. Just giving you a little hint for those who do not, that diabetes is deficiency of insulin that is produced by the pancreas. So pancreas produces insulin. Insulin is moving in the body. When you eat glucose, this insulin is going to bind with this glucose and take it to different cells of the body. So the cells get the glucose and you feel good and healthy. For some reason, if the pancreas is not able to release this insulin, is not able to produce this insulin, what will happen then? The glucose is going to come in through your diet. But who is going to take the glucose to different part of the cells in the body? It doesn't have the carrier molecule in the form of insulin. That is the reason you take oral tablets which are going to help to carry all these glucose molecules to different parts of the cells. And similarly, if the oral is not effective in juvenile diabetics, you take injectable insulin, which is going to help carry the glucose molecules to different parts of the body. For small little children, sometimes it hurts to see children who are 3-year-old, 4-year-old, 5-year-old, and they have to take their injections of insulin morning and evening. Imagine all the shots, they take it on their thighs, they take it on their hands, and you have these small needle marks all over the body, and they are such young children. And these biosensors have been a boon for these little children because they have devised a small pump, and very fondly they call it the artificial pancreas, because this small pump has the insulin inside and it has the biosensor inside. So this is kept under the flap of the skin. Imagine this, just close your eyes and imagine you have a small pump which has insulin. You flap open the skin, put the pump inside and put the skin back. Then what does the pump with the insulin do inside? It has this small little biosensor which has an electrode which will monitor the glucose that is present in the blood. And once the level of glucose goes down or high, let us say it goes high. If the glucose goes high, the electrode immediately knows that now the glucose is going high, the small kid is going to be hyperglycemic. So it will start releasing insulin in the required quantum. It has been made in such a way that if your sugar is 300, it knows how much insulin to release. If the sugar is 400, it knows how much insulin is to be released. So this is a blessing in disguise to all juvenile diabetics. It is very, very expensive. We cannot forget this, that all these devices definitely come with a cost. But I am confident with the kind of scientists we have around, we will very soon have something which is more economical and which can be used by everybody. But at the moment, it is, yes, very expensive, but it is going to be wonderful for all the children who are juvenile diabetic and who have to take injections morning and evening. So this has been a big help. There are n number of applications, but we cannot discuss it at this moment. Just remember that these are sensors which are used for biological purposes, and we are using it and we are exploiting it to the greatest in the medical, in vitro and in vivo faculties.
Thank you, Bhavini, for explaining the use of biosensors in medical field, and it was very well explained. Now, apart from medical aspect, biosensors can be used in many other industries. Like you can use these biosensors in food industries. Suppose you want to know the composition of the food, how much sugar is present, how much fatty, what is the amount of fatty acids, or even what are the amounts of vitamins. You know that for vitamins, if you want to analyze, then you are using bioassay, which is a tedious procedure. So if you just have a biosensor which can straight away check the presence of riboflavin, that will make the things very easy. Similarly, you can detect the presence of toxic substances like mycotoxin. Even you can detect the microbial load, as I have already talked about the enzymes, optical biosensor, luciferase based optical biosensor, and I explained you how it can be used to detect the microbial load, and it also helps in determination of levels of toxic substances before and after food processing. Biosensors, they can also be used for agriculture. Very well, you can very easily identify viral, fungal or bacterial diseases, especially if the fungal spores are present in the atmosphere. They can be very easily detected by using the immunobiosensors, where you can have the antigens over the antigens of those pathogens immobilized on the biosensors, and naturally the pathogens, they will be attracted towards those antigens, and thereby they can be sensed, their presence can be detected, right? So similarly in the fields, if you have used pesticides or fungicides, so you can use the microbial biosensors to detect the presence of those substances, those chemicals, and you can also find out whether the remaining pesticides, whether they are biodegradable or they are not, right? So that is how in agriculture they are responsible for helping us. Similarly, they are also very important in detecting the environment pollutant. About that we have already talked. But just to give you a simple example, you know that BOD is very important, right? So if you have to check the level of BOD, if you want to know the value of BOD, then you all know that the procedure requires 5 days. It is a very long procedure. So if you are using microbial sensors, like if you use a special type of yeast, Trichosporon cutaneum, it can give you the value of BOD within 30 minutes instead of 5 days. So you can very well imagine how interesting and how easy things will become. Similarly, if you want to study the mutagenic or the carcinogenic effect of some chemical, so what you have to do is just take the immobilized S typhimurium or Bacillus subtilis, and then along with oxygen electrode, you can very well detect the mutagenicity or carcinogenicity of various chemicals. You can use portable pesticide monitors to measure the degree of contamination in aquatic environment. Then of course biosensors, they can be used in industries also. Now it can be any type of industry, it can be pharmaceutical or it can be petroleum industry, construction industry, etc. Now, very interesting thing is, which initially, in mining industries, parrots were used. So, basic thing was that if toxic gases are there in the mines, then these parrots are very sensitive to the concentration of the toxic gases. So, immediately they will recognize those toxic gases and the parrot will die. And the moment that parrot dies in the cage, it is a clear indication that, okay, this place is not safe, you should leave the mine. Right. So, that is how the biosensor, actually the concept of biosensing came, right. So, similarly such biosensors can be used for alerting the workers who are working in the mines about the toxic gases. You can also, in fermentation industries, suppose you want to know the temperature variation, so that can be, you can use the thermistors, or if you want to use the pH fluctuations in the pH, so pH electrodes can be used, dissolved oxygen can be measured. So, all type of biosensors are available which can detect the physical parameters as well as chemical parameters of the process which is going on. Right. So, these biosensors, they are very important in the industries. Then we have application of these biosensors in the forensic medicines. Right. If you remember, when we were talking about the piezometric biosensors, at that time I told you that they are very commonly used to detect the presence of cocaine. What we do is that piezometric crystals, they will have the antibodies of cocaine, right, and they are absorbed over the surface. And then if in the atmosphere that cocaine is present, then it will naturally absorb to the specific antibody and oscillations will change. So, you will come to know that, okay, cocaine is present in this environment. Similarly, if you have let us say anthrax is there is some sort of terrorist attack, right. So, if anthrax spores are there in the atmosphere, so you can very well use the immunobiosensors. Antigens are there. If in the atmosphere anthrax organisms are there, they will naturally they will be absorbed over the surface of the biosensor and there will be clear indication. So, in forensics, even you can check the presence of poisonous material like cyanide, whether it is there, cyanide poisoning is there or not. So, it can be very easily checked by using specific biosensors. Then biosensors are also being used in military and defense industries. You know that there can be either chemical warfare in the industries. We know about the weapons, right? That is another aspect only. But if there is chemical warfare or if there is biological warfare, then it can be very well detected by using the biosensors. As I have already said, that suppose there is biological warfare. So, you can very well check the presence of anthrax spore or any other biological, any other pathogen if it is present, especially airborne pathogens which are present. So, straight away you can keep the nano biosensors and which can detect the presence, and even if there is mustard gas or nerve gas, it can be very easily detected. So, that is the use of biosensors, and these are the wearable biosensors. You can wear the biosensor in your ring and nobody will notice it, and you will come to know you will get the information. This is the smart shirt you can wear having the biosensor and you will get all the information.
Spice can wear it.
Spice can wear it, and it is very interesting. That is why it is known as smart shirt. Now, these biosensors, they have got lots of great future about which my friend Bhavini will discuss.
So, at the end, one more very fascinating application of biosensors. As we know, cancer is one dreaded disease. Okay, so it is so fascinating that if a patient is diagnosed of cancer, you have this small chip of biosensor which is specific to a cancer. Let us say it is ovarian cancer. It has specific metabolites. It has specific cells which are very difficult to see with naked eye inside of the body. So, if once you are diagnosed with the cancer and you are on treatment, and if you have that small little specific biosensor inside of your body, which is coated with specific biosensors, which will immediately sense if there is increase of the tumor, if the tumor is spreading anywhere else in the body, and it will give you a sensing physical reaction. In that case, it is very, very simple for the doctors. For the patient himself, he will immediately know or he will immediately sense if there is some problem, and that could help save a lot of lives, lot of money, lot of mental and physical trauma to the patient and the family. I am so very confident that all of you students must have sensed thoroughly what is biosensors. But still, we teachers are always available for you, and it is difficult for us to show you all the applications. But we have put forward on our last slide one of the email IDs so that you could very easily note it down for any further questions. You could note down the email ID so that if you have any questions regarding biosensors, its applications, you have any confusion, take your own time, both of us are available, just put in a mail and we will immediately revert back to you. Thank you so much for having us.
Thank you so much.